Worshipping in spirit and in truth. Glory to the faithful one, Jesus Christ, God's Son. All creation gives you praise. You alone are truly great. God who reigns for eternity, yeah. God is great, and His grace fills the earth, fills the heavens, and Your name will be praised all the world. God is great, sing His praise on the earth, all the heavens, 'cause we're living for the glory of Your name.
இருந்துட்டு போடு சார். ஆமா அவ்வளவுதான். அதுக்கு நம்ம
Science and technology revolutionize our lives, but memory and tradition frame our response. Let me now invite Dr. B. Divya, Assistant Professor, to welcome the gathering.
Good afternoon to one and all present here. I'm delighted to offer a hearty and warm welcome in this beautiful day. First, I welcome the chief guest of this program, Dr. Satish Kumar, IBS, Senior Postdoctoral Researcher, Post-Tech, South Korea. Let me share a few words about him. He did his BSc in Kamaraj College, Thoothukudi, MSc in Manoormaniam Sundaranath University, Tamilnadu. He completed his PhD in the University of Hyderabad. Now, he is a senior postdoctoral researcher in South Korea. His list of achievements is enormous, so let me say a few. He has qualified SET exam in the year 2012, and he has cleared NET exam LS for 2 times, and he also cleared a CSIR JRF, and he has cleared GATE exam for 2 times. And he presented various papers in various conferences, and he has published many papers in reputed journals, and also published a chapter in a book entitled Functionalization of Carbon Nanomaterials for Environmental Application. So really, we are fortunate to have you here, sir, as resource person today. I welcome you, sir. I also welcome our secretary, Reverend Sister Flora Mary, our principal, Dr. Sister A.Z. Luziaros, our HOD, Mrs. Rani Jai Mary, and all the staff members of our department. I also welcome the teachers and the professors from various parts of our country. So once again, I welcome you all.
Thank you, Divya, for your cordial words of welcome. Achievers never expose themselves, but their achievements expose them. Dear participants, now it's the time to take the photo session. Kindly participants, switch on your video to make your presence a memorable one.
Thank you, ma'am.
Thank you, participants. The object of education is to prepare the young to educate themselves throughout their lives, says Robert. Let me now invite our speaker of the day, Dr. P. Pon Satish Kumar, IBS, Senior Postdoc Researcher, Institute of Basic Science, Post-Tech, South Korea, to deliver the lecture on the topic Gene Delivery Challenges, Advancement and Applications. The session is yours, sir.
Could you able to view the presentation?
Yes, sir.
Yeah, right. Good evening, one and all. So today I'll be presenting our gene delivery challenges, advancement, and application. So the human mankind wants to be away from any diseases, but still once in a while they will be crossing across one of such diseases. So depends upon the disease conditions and exposure. There are many treatments available. So they are from antibiotics or surgery or anti-cancer. There are so many treatments depending upon the condition of the diseases. So there is one rare condition disease which is called as genetic disorders or genetic diseases. So these are generally caused by the deletion or mutation of the genes, so which result in the irregular production of the proteins or malproduction of proteins, which result in many pathologic conditions, which include cystic fibrosis, Marfan syndrome, and so on. So one of the advanced treatment for these genetic diseases is the gene delivery or gene therapy systems. So this basically the gene therapy involves the delivery of genetic material which can compensate the abnormal gene or that can make beneficial protein inside the cell so the condition can be treated better. So but as like we take the medicine either by intravenous or intramuscular or orally, we cannot deliver these kind of materials directly into our body. So why we cannot directly deliver like that? So these DNA molecules are hydrophilic in structure and they are large size, and so they cannot easily penetrate into the cells, and they can be easily fragmented by the nuclease enzymes when they enter into the physiological condition. So this is maybe due to the the both endo and exonuclease activities inside the plasma. For example, if an active DNA is introduced inside the body, then its lifetime will be like 1.2 to 21 minutes, which is not much suitable for making the gene delivery. All right. So what will be the solution for this? So the solution is you have to protect the DNA, and you have and then uh, we need to deliver it to the particular cell. So for that, the role of the gene delivery carriers comes here, and so ideal gene delivery system should meet some criteria. So what are the criteria? There they should able to interact with the DNA, but not permanent interaction. It is kind of electrostatic interaction should be there, and they should able to carry the DNA inside the physiological condition without any damage. And these carriers should not cause any adverse effects. So these are the ideal characteristics for any gene delivery system. All right. So these gene delivery systems are mainly classified into viral and non-viral carriers. Generally, the viral carriers or the viruses, which are modified viruses, are um, they have the replication deficiency. So in such way, it is modified, and these uh, therapeutic genes are loaded inside these particular viruses, and then when it is treated with the cell, then it delivers the particular gene inside the cell, and which produces the gene therapy. And there are some systems developed so far are adenovirus system, retroviral system, and lentiviral system, and poxviral systems. So, all right. So, although these uh, virus materials are very good for the transfection of this genetic material inside the body, that means it can easily go and deliver. But they have some severe disadvantages, which are like they have mainly this post oncological effect and immunological side adverse effects. So to overcome this, there are some other methods developed, which are mainly physical method and chemical method. So the physical method involves the needle injection or ballistic injection. So these two involves the utilization of a gene gun, so which can directly deliver it into the cell, and the sonophoration, photophoration, electrophoration. So these involves utilization of sonication and laser and electrical pulse. So they when they when they apply it to the cell, then the membrane gets some kind of destabilization, and with that, when the DNA is present in that condition, then they will be delivered inside the cell. All right. So whereas in the case of chemical method, there are different synthetic and natural biodegradable particles which are able to carry these genetic material inside the body. All right. So these involves cationic polymers, dendrimers, lipid nanoparticles, and peptides. So these are the some representative figures for such a delivery system. So in the upcoming slides, I will give you some deep introduction and application of this delivery system. All right. So when we go into the chemical delivery systems, or so these involves uh, it can able to pack the DNA with its structure, and it can take this material into the cell and make the transfection possible, and which in turn can affect the gene delivery. Using all right. All right. So these particular materials involves, as I said, these are the kind of example for the polymeric lipid micelle, and this is the protein nanoparticles, and this is dendrimer, and this is kind of liposomal. So the upcoming slide I will explain one by one. So when you have these non-viral vectors, especially the chemical method of non-viral vectors, so they basically involve three type of interaction with the genetic material. So when you treat this DNA material to this chemical carriers, so they make electrostatic interaction, and so that means a kind of hydrogen bonding interaction or ionic interaction, and then they make the encapsulation within the formed structure, which is shown as shown here, and absorption of biodegradable nano or micropores, so that makes the the surface encapsulation of the genetic material, and it goes and for delivery. So in the so I'll explain in detail, so with good examples. So I'll start with the non-viral vectors with cationic polymer. So this cationic polymer is nothing but linear polyethylene imine. So if you see this structure, so there are many this kind of amines are present in its structure. All right, so they can easily make the hydrogen bonding or ionic bonding with the negatively charged this genetic material, and they can carry over this transfection possible. All right, so when you come up with the simple polyethylamine, then there are many adverse effects. So the researchers makes some kind of polishing of it. That means it so they attach some kind of other polishing materials like folic acid or some kind of polyethylene glycol. So they will be conjugated. Then when we go for further gene delivery, it will be very efficient, and the cytotoxicity level will be decreased. So one such example is the folic acid decorated polyethyl glycol polyethylamine copolymer. So what happens here is the uh, folic acid is conjugated with the corresponding polyethylamine by means of some covalent modification. And when you treat this one with the DNA, so it makes some interaction with the DNA and forms the corresponding nano carriers. When it is delivered to the cells, then they take into the cell, and inside the cell, the DNA will be released, and then the transfection happens. And then finally, the gene is delivered to the nucleus, and the gene therapy can be possible with that. So further, so if you look at this particular molecule, so here they have utilized the polyethylamine, which is conjugated with the cysteine. So this particular system is kind of stimuli responsive. So what kind of stimuli? So that is GSH stimuli responsive, glutathione responsive. So when this particular material is made, and then it will be treated with the DNA and make some kind of nanocarrier like this, and this nanocarrier is able to form, the protect the particular DNA, and it has a many positive charge on its surface, so it can easily go into the cell, and then further, so the endoplasmic, after the endoplasmic escape and inside the cell, that glutathiones makes this SS bond disruption, and which immediately releases the DNA and the genetic material. Then it will be going into the nucleus, and that produces the gene expression, which results in the gene therapy. So in this particular system, it's shown very higher buffer capacity. So which means when it goes into the the biological environment, there the pH of the solution or the pH of the system will not be altered. Otherwise, when it goes into the biological system, the particular pH altered on the surface, then the degradation of the DNA or the genetic material happens. And this particular material, it makes the large enhance the interaction with the DNA as well. So they can easily hold the DNA and they decrease cytotoxicity and enhance gene transfection efficiency. That means the delivery of the genetic material inside the nucleus. All right, so this the next particular material, which involves the similar, the polyethylamine, which is particularly branched. But in this case, they particular in this particular case, it is treated with some hyaluronic acid derivative, so which is conjugated with the uh, thiobutyric acid, which is also glutathione responsive material. So what generally happened in this case is the polyethylamine is treated with the DNA, so they form some kind of DNA PA binary complex, and then which is further treated with the hyaluronic acid conjugated dibutyric acid, so which in turn gives very high protection for the particular genetic material. And also they because of their polaric nature, they can easily penetrate to the particular cell and makes the transfection possible. So in this particular material, so they found that the transfection efficiency is increased by 14-fold. So with this, I move to the next non-viral vector, which is called as dendrimers. So dendrimers are a kind of polymer only, but they have the branch repeating units. For example, if you look at this particular structure, this is the first generation dendrimer, and this is second generation dendrimer, and this is third generation dendrimer like structure. All right. So this particular dendrimers have very good advantages. So they have a good surface of uh, the amines, and they can easily interact with the genetic material. And there are some cavities. So these cavities can be accumulated by some other additional, the carrier supporters. All right. So with this uh, dendrimer, there are many applications have been carried out. I will discuss one by one. So in this particular case, uh, the PM, yeah, AM, all right. So this particular dendrimer is treated with HMGB1. So that means the nuclear locating sequence of some kind of peptide. So what it do is like when it is introduced into the nucleus, that directs the movement of the particular material towards the nucleus. All right. So when these three are mixed together, they forms a ternary complex, and this ternary complex further treated with the folic acid conjugated polyethylene glycan, so which in turn can give a good protection and decrease the cytotoxicity. So further, so they form multi-component material which then treated with the cell, so which gives the good transfection inside the cell, and which is evident from the corresponding gene expression which is observed in that particular system. So as expected, this system showed less toxic effect and enhanced gene transfection. So this is another type of dendrimers which have the arginine system on its surface. So this is the particular dendrimer, and when this dendrimer interacting with some kind of inorganic nanoparticle, then they make superstructure. Then they can, they can offer good transfection inside the cell. All right. So how this is happening? So the dendrimer, which is having some kind of the coordinating groups, and these coordinating groups can interact with the inorganic nanostructures, and so they form the superstructures. And these superstructures, when it is treated with the DNA, so they can easily hold the DNA, and when it goes into the cell, and so they can easily carry out the transfection and cell penetration because of this arginine moiety. And when it goes into the cell after the endoplasmic escape, and they releases the DNA, and the transfection happens properly. And in this case, because of this arginine unit and the particular the inorganic nano carrier. So they found that 50,004 increase in the transfection ratio, and they found the low cytotoxicity as well as in the serum resistance in gene delivery. And in this particular case, the interesting thing is like this transfection can be monitored by the fluorescence, as this the inorganic component or fluorescence enough to show off where the particular material is moving inside the cell. This can be achieved by the fluorescent microscopes. Further, the similar system can be utilized by the stimuli responsive carriers. These stimuli responsive carriers are made by the particular dendrimer. That means the polyamidoamines are treated with particular butyric acid, diobutyric acid. So these diobutyric acids are like having the SS bond. So these are these are sensitive to the glutathione. So when the glutathione is present, they will be cleaved. All right. So in with keeping in this mind, they designed this particular nanocarrier, and these nanocarriers are formed by the covalent interaction between this butyric acid and the nearby polyamidoamine. All right. So when this particular dendrimer are gathered by the covalent interaction, further it is treated with the genetic material, and when this genetic material is interacting with the uh, these dendrimers, they make very tight bonding, and further during the gene delivery, so it goes into the cell after the endoplasmic escape. It is exposed to the GSK glutathione, and when the glutathione offers this kind of breakaging between these dendrimers, polyamidoamine dendrimers, then they releases the corresponding genetic material, and which leads to the transfection and into the nucleus and gives the expression over there. All right. So this is the concept behind this particular system. So with this, I move to non-viral vectors based on the peptides. Some of the peptides which are derived from the viruses are found to make the gene delivery efficiently high. All right. So, but they have some disadvantages. So the other peptides are focused for the particular development of a particular system. So in this particular system, a peptide is treated with the particular genetic material, so which can easily form this so all the peptide have this acid group and the amine group, free amine group, so they can interact with the corresponding genetic material, and they can form the uh, the nanoparticles. And similar system, if it is treated with the polyethylene glycolated peptide, that means the peptide which is conjugated with the polyethylene glycol group. So why we have to do this? This is because we can protect the DNA. In one way, it is protect the DNA. Other way, it will decrease the cytotoxicity of the material. All right. So when these three are mixed together, it produces the polyethylene glycolated genetic material nanoparticle. So when this material is made and then they are treated with a particular cell, and then it will be going inside the cell and makes the transfection very feasible. So this particular material shows the the less cytotoxicity as well as the higher transfection when compared to the only the simple peptide and the genetic material. So this is another type of system in which this cell penetrating peptides are conjugated with the particular genetic material. That means you have the peptide, then that is conjugated with the particular genetic material to be delivered by the SS bond. So as we already know that SS bond is sensible to the GSH. When it is exposed to this cell, then it will be easily releasing the genetic material inside the body. All right. So, but after making the conjugator with the CPP, and then it is then treated with the peptide, which is conjugated with the polyethylene glycol and folate. So this folate group are, so they can drive this particular material towards the uh, uh, cancerous cells. So instead of going to every cell, then they will directly go to the cancerous cell. And inside, so when they goes into the cancerous cell, and at that particular pH, they releases this conjugated genetic material with the CPP. And once it goes into the cell, and because of the presence of the GS height, these SS bonds are cleaved and genetic materials are released. So once it goes into the cells, then it reaches the nucleus and produces the gene expression. So these gene expression, the mRNA and protein levels indicate the constructed vesicle exhibited enhanced cell cancer cell apoptosis. That means the targeted delivery enhances the the cancerous cell deletion. So we will. So with this, I move to the another important type of non-viral vector delivery system. So which is using the lipids. So this is the one of the promising non-viral vectors. And recently in the mRNA vaccines, these particular type of non-viral vectors are used. So coming to its structure, so this structure will have cationic lipid. In case of cationic lipids, it has the head pod and a connecting pod and the tail pod. So the head pod will be a cation. So most of the time it will be a particular quaternary ammonium salt. And in case of ionizable lipids, so that will be the amine pod. So that can be, when it is interacting with the kind of protonate acidic environment, they will be ionized. All right. So there are some other type of lipids as well. So which involves the phospholipids and cholesterol. So going to its application. So when we treat the particular the lipids with the DNA in the water. So when they mix together, they form a vesicle like this. So finally, they end up with the highly protected DNA vesicles. And these DNA vesicles, when they enter inside the cells, they releases this DNA inside and produces the cell expression which is needed for the gene delivery. So in this particular system, so here is a particular example, and in this system, what they have performed is like this is the cationic pod, and this is the tail pod. So in the cationic pod, instead of cationic pod, what they have used is a peptide system. So the peptide system consists of lysine, arginine, and glycine. So this peptide system act as the head pod, and whereas this all other tails are remain same. So they have utilized different sort of the peptides by dipeptide and tripeptides, and they found that the KGG peptide, when it is attached to the head pod, and the transfection and the particular cytotoxic activity is very low compared to the other system. So this is because of this particular peptide arrangement makes the good interaction initially, and that makes the transfection very high. So with this, I move to the next one. So in this case, the tail pod they have utilized the alkyne, whereas the head pod remained at the same. So what generally happens is, so when you treat this one with the genetic material and they form vesicle like this, when these vesicles are treated with the cell, so they produces the corresponding gene expression, which resulted in the corresponding gene therapy. So with this, I move to the successful applications of this uh, gene delivery system. So uh, this recently, the SARS-CoV-2 or this COVID vaccines are made using this kind of lipid uh, delivery gene delivery systems. So which involves the carrying of the mRNA, and so when this uh, mRNA is encapsulated inside these lipids, and when it is inserted to us, and then it produces the transfection and inside the body. So when it reaches the nucleuses, the nucleuses inside the nucleus, it makes the gene expression which produces spike protein. So when the spike protein is released to the environment and some antigens are produced, and then further, the physiological memory makes the uh, what do you call the vaccination possible. All right. So with this, I conclude, and I thank the organizing committee of St. Mary's College for a good chance for discussing the gene delivery systems. And thank you, one and all, for your kind attention.
Thank you, sir. Now it's time for open discussion. Kindly participants unmute your microphone and clarify your doubts. Dear participants, thank you, sir, for your wonderful lecture you have given.
Yes, ma'am.
Through your lecture, several information about more things have been come to our notice. Thank you, sir. Thankfulness is the beginning of gratitude, and gratitude is the completion of thankfulness. Now I invite Dr. A. Lakshmi to propose the vote of thanks.
Gratitude is not in the words, but in the heart which expresses it. A very good evening to one and all connected here through this virtual platform. It's my pleasant privilege to propose vote of thanks in the fourth day of this online FDP program. First and foremost, I thank Lord Almighty for showering His blessings to complete this session in a fruitful manner. My heart fills with the lots of gratitude for our resource person, Dr. P. Pon Satish Kumar, IBS Senior Postdoc Researcher, Institute of Basic Science, Post-Tech, South Korea. Thank you, sir, for enriching us through your most informative lecture on gene delivery challenges, advancements, and applications. Really, it's very interesting to know different non-viral vectors, their transfection and applications. I thank our Secretary, Reverend Sister Flora Mary, our Principal, Reverend Dr. Sister A.S.J. Lucia Rose, Deputy Principal, Reverend Dr. Sister Colanda Chris, for their constant support and encouragement. I take this opportunity to thank Dr. Sister Aurokya Genetius Alphonse, the overall coordinator for DBT Star College Scheme, and Ms. S. Rani Jayamary, Head Department of Chemistry, for their valuable contribution and guidance. My special thanks to the technical team for their continuous support. Last but not the least, I thank all the enthusiastic participants. Finally, I thank one and all. Thank you.
Thank you, Lakshmi. Dear participants, Kindly fill the feedback form posted in the chat. Dear participants, kindly fill the feedback form posted in the chat box. Tomorrow, the Day 5 program will start at 10 a.m. Thank you all for your support and concern.
Satisha, thank you so much, sir.
Yeah, yeah. It's my
Thank you for
pleasure.
accepting our invitation, sir.
Yes, yes, yes. It's my pleasure.
Thank you, sir.
Yeah, yeah. All right.
You can leave the meeting, sir, now.
Yeah, yeah.